Oh, sorry. Good morning. I just started the chat, so sign in on the chat. Just wanted to know, how was the presentation yesterday? Let me get some feedback on that. Okay, it's 10:30. Did anyone hear my question? Okay. Here's my question. How was the presentation? How were the presentations yesterday evening? Let me hear what your feedback is on that. Okay, I'm getting responses now. But did you hear my question on the Hangout? Yes, Charlotte, the test on Monday covers chapters 12, 13, and 14. Okay, let's get started. Let's see if anyone is in the Clementine. Okay, Clementine, I see that you're in poll everywhere, so that you're here. Okay, so I am acknowledging that I see you. Okay, I want to start with a couple questions relating to last lecture. I am going to share my screen and have those questions posted. Let me pull up the slide. So here's the question. Just a minute, sorry. So the question is, which protein will be bound to the lac operon DNA of E. coli grown in glucose? So which protein will be bound to the lac operon of E. coli grown in glucose? And do not include RNA polymerase as an answer. And I will look for your answers on Facebook. Okay. And Clementine, you could go ahead and respond via the poll. Okay, Erica, yes, repressor. Let me pull this up. Okay, let's look at it again to see why it's not the CRP cyclic AMP complex. So the question again, which protein will be bound to the lac operon DNA of E. coli grown in glucose? When glucose is present, there is no need to produce the genes necessary for utilizing lactose. So the repressor protein will bind to will bind to the operator site. When glucose is present, when glucose is present, it prevents that complex, that cyclic AMP CRP complex from forming. And if that complex is not formed, then transcription is not enhanced. So transcription of the LAC genes will not be enhanced, because that CRP cyclic AMP complex is necessary to facilitate binding of RNA polymerase to the promoter site. Okay, so when cells are grown in glucose, when E. coli are grown in glucose, you will have the repressor protein binding instead. And I'm going to answer that on the chat. When okay, second question. Which protein will be bound to the lac operon of E. coli grown in lactose? So when cells are grown in lactose, which protein is bound? Again, do not include RNA polymerase as an answer. Yes, this one is CRP, cyclic AMP complex, yes. Okay, third question. I will put that up now. Let's see what Zach is saying. Zach is typing here. Yes. So if lactose is present, there is a need to utilize to produce the genes necessary for utilizing lactose, and so it would be important for the genes to be transcribed, and for transcription to occur, you need RNA polymerase binding. But to enhance binding of RNA polymerase, you need the CRP cyclic AMP complex. So that would bind, and that complex will enhance the binding of RNA polymerase to the promoter site. Last question. Okay, last question. Which protein will be bound to the lac operon when cells are grown in both glucose and lactose? So you have both glucose and lactose present. Which protein will be bound to the which protein will be bound to the operon? Lactose and glucose are both present. Okay, I'll wait for your answer. Okay, Clementine, I just saw your answer. Yes, you're correct. The complex of the cyclic AMP and regulatory protein, that was correct. For the second question, that was correct for the second question. Okay. Okay, I see a lot of people are saying repressor protein. It's not correct, because if lactose is present, lactose will bind to the repressor protein. And when lactose is bound to the repressor protein, the repressor is unable to bind to the operator site. So the correct answer is not repressor. Let's try again. Yes, it does favor glucose, Zach. So if both are present, both glucose and lactose are present, we know that the repressor protein will not be bound. Because the repressor will be bound to lactose. And Clementine, you can answer in the poll, okay? No one? No one answering? Let's see. I'll give you 50 more, well, 30 seconds. Okay. Okay, so if both glucose and lactose are present, no protein will be bound to the lac operon. Because number one, if lactose is present, we know lactose will bind to the repressor protein, and that will prevent the repressor from binding to the operator site. If glucose is present, then the cyclic AMP-CRP complex cannot be formed, and hence that will not be bound to the lac operon. So the correct answer is no protein will be bound if both lactose and glucose are present. Okay? So that's it. Let's see what Clementine said. None of them. Very good. Look, very good, Clementine. You got the answer correct. Very good. No repressor will be bound. Okay. And as a result, you will have low levels of transcription of those genes, but it just won't be enhanced because the cyclic AMP-CRP complex will not be there to enhance transcription of the genes of the lac operon, right? Okay, so let's move on to today's lecture. Today, let's see, we're gonna cover. So last couple of days we have focused on the lac operon, and we said that that is an example of a catabolite operon. So catabolism refers to the breakdown of molecules, and that is one side of metabolism. Metabolism also involves building up or making or synthesis of nutrient requirements. And a different type of regulation is used in anabolism, which is the buildup or synthesis of molecules. The tryptophan repressor represents an example of an anabolic operon, or an operon that uses, well, anabolic operon. Okay, so let's see what the trip operon looks like. Let's see here. Do I have it written? Yeah, here's a trip op, the tryptophan operon. The tryptophan operon represents, again, an anabolic operon, anabolic synthesis. And let's see let's look at the structure of the operon, and then we'll look at how it's regulated. Okay, here's the structure of the operon. Okay, so the tryptophan operon has structural genes, and they're labeled TRPE, D, C, B, and A. These are similar to the LAC-Z, Y, and A genes. Those are structural genes. And if you recall, structural genes are genes that encode the information for making protein. Okay, so tryptophan, the trip operon, has 5 structural genes. That's TRPE, D, C, B, and A. Adjacent or upstream of the first structural gene, you have what is known as your leader sequence, a leader sequence, and we'll see how important that sequence is in regulating the synthesis of, or regulating expressions of the structural genes found on this operon. So there's a leader sequence, and then upstream of the leader sequence, you have your operator site, and then upstream of that, you have a promoter site. Okay, so trip operon, you have promoter, operator, leader sequence, and your structural gene. So all anabolic operons, all operons that are involved in the synthesis of nutrients or any molecule would contain this leader sequence. Within the leader sequence, there's a significant site known as the attenuation site. The attenuation site, and this is the region that plays a role in regulating transcription. So attenuation refers to a type of regulation that involves both transcription and translation. It involves both transcription and translation. So we'll expand on that when we get to that section. Okay. Okay. So bear in mind the structure here. So here's your operator site. If the operator is free, similar to what happens with the LAC operon, when the operator, if there's the repressor binds to the operator. If the repressor binds to the operator, then no transcription will occur. Okay? If the repressor doesn't bind, then you have transcription occurring. Okay, so it says repressor cannot bind, then transcription will occur. Okay, let's move on. Okay, so with these anabolic operons, as the end product, which is tryptophan, acts as a co-repressor of the operon. So in addition to having a repressor that binds the operator site, the end product, which is tryptophan, acts as a co-repressor of the operon. So how does that work? When it shows you here in this figure, you see the promoter, operator site, your leader sequence. Tryptophan acts as a co-repressor to the repressor of the operon. The repressor of the operon is known as TRPR. So TRPR binds to the operator site, but it binds to the operator site in combination with tryptophan. So both of these are necessary to bind to the operator site. And so tryptophan acts as a co-repressor. It says here, binding of the tryptophan to the TRP repressor allows the TRP repressor to bind to the operator. So unless tryptophan is bound to the operator, unless tryptophan is bound to the repressor, it is unable to bind to the operator. So they must be joined in order to bind to this operator site. So it says binding of tryptophan to the repressor allows the repressor to bind to the operator and inhibit transcription of the structural genes, of the 5 structural genes. In the absence of tryptophan, the repressor is unable to bind to the operator. So when there's no tryptophan present, then the operator site remains free, RNA polymerase is able to bind to the promoter site and allow for transcription of the structural genes. These structural genes are necessary for synthesizing tryptophan. Okay, so these genes are necessary for making tryptophan. Unlike the lac operon, the genes, the structural genes, were necessary for breaking down lactose. Okay, so only when lactose was present, these genes would be transcribed. On the other hand, with the trip operon, like all other anabolic type operons, if tryptophan is present or the end product is present, then there's no need for the structural genes to be turned on, and as a result, the structural genes will be turned, transcription of the structural genes would be inhibited by the end product, in this case tryptophan, binding to the repressor protein, and that complex will then bind to the operator site and prevent transcription of the structural genes. Okay, let me see if there are any questions, and we'll move on. I'm going to check for questions. Okay, no questions so far. Okay, so no questions. Let's move on. Okay, so let's see how this regulation works on a molecular level, closer level. Okay, let's see the role of each of these components in regulating transcription. So we see the role that tryptophan plays. But if you recall, we said that the trip operon has this leader sequence, and within the leader sequence is what is known as an attenuation site, and this attenuation plays a key role in regulating transcription from operons that are involved in synthesis. Okay, so the leader sequence has the ability to form two different types of configurations. So when the DNA of the leader sequence is transcribed, it has the ability to form two different conformations, and the conformation form determines whether or not transcription of the structural genes will continue. Okay, so the leader sequence being transcribed determine the formation, I should say, the formation of the transcribed leader sequence determines whether or not the downstream structural genes will be transcribed. Okay, so these are the two possible conformations of the leader sequence that could be formed. If you have a 3-4 stem loop that is formed, that 3-4 stem loop is known as a terminator. That configuration will prevent transcription of the downstream structural genes. Okay, so here the leader sequence, I should point out, has 4 regions. There's one region known as 1, 2, 3, and 4. These are the 4 regions. If there's a stem loop formed between regions 3 and 4, as in this case, if the stem loop forms between regions 3 and 4, then transcription of the downstream structural genes. If this if the stem loop forms between regions 2 and 3, that is, if 2 and 3 binds together to form a stem loop configuration, that creates what is known as an anti-terminator, and that anti-terminator would allow full-length transcription of the structural genes. So let's see what happens when tryptophan is present and when tryptophan is absent. Okay? Okay. When tryptophan is present, when tryptophan is present, there is no need for the structural genes to be formed. But this is what happens. So when tryptophan is present, we have this is our leader sequence that was transcribed, and you can see the 4 regions, 3 and 4. Leader sequence is transcribed, and if you recall, if you remember that in prokaryotes, transcription and translation are coupled. Okay, so once you have the transcription begin, once transcription has begun and there is some messenger RNA available, the ribosomes could be immediately bind to that transcript and begin synthesis, even before begin protein synthesis, and that can occur even before synthesis of the mRNA is completed. So transcription and translation are coupled; they are occurring together. Almost simultaneously. So here's a leader sequence that was transcribed. That's a leader sequence. If tryptophan is present, then ribosomes will bind to that leader sequence. And when it encounters on the leader sequence in region 1, you have the codons for tryptophan. So when the ribosome approaches these codons for tryptophan, it will be able to move along that region, because tryptophan being present, it would allow charged tRNAs would be available. So you'll have tRNAs that are charged with the tryptophan amino acid will be available and allow for translation to begin. Okay, again, translation and transcription are coupled. So here we have a ribosome encountering a trip codon that would allow charged tRNAs charged with the tryptophan amino acid would be available, and that would allow the ribosome to move quickly past region 1 and allow the formation of this 3-4 stem loop structure. If the 3-4 stem loop structure forms, that prevents further transcription of the structural genes. Okay, so the structural genes will not be synthesized. That's because the ribosome moving past region 1 allows for the 3-4 configuration to form. If tryptophan is not present, then the ribosome, when it encounters a trip codon, will stall. It won't be able to move past region 1 because there are no charged tRNAs with the tryptophan added to it will be available. So none of these charged tRNA trips will be available, and so the ribosome will become stalled at the trip codons. That allows for the 2-3 stem loop to form, and this 2-3 stem loop, we know, is an anti-terminator of transcription, and so as a result, transcription of the structural genes will continue. Okay, so I said a lot, and I'm going to check to see if there are any questions to what I just talked about. Clementine, go ahead and ask your questions in the poll if you have any. Okay, Zach, I see your question. Is this sort of like negative feedback, too much tryptophan, the tryptophan will bind? And yes, Zach, that is correct. So any questions regarding the attenuation? So what I described was attenuation. The response, let me define it as your text defines attenuation. It's the control of gene expression by the RNA leader-mediated premature termination of transcription. So attenuation, control of gene expression by leader-mediated premature termination of transcription. So attenuation is the premature termination of transcription via this leader sequence. All right. Any questions? If not, we'll move on. Let's look at other ways of regulating gene expression. So the regulation via attenuation is an example of an RNA-mediated mechanism of gene regulation. So the RNA itself is involved in regulating gene expression. The conformation that the RNA forms, which is a leader sequence, the transcribed leader sequence, that conformation determines how, or is involved, well, determines how the genes are, whether or not the genes are expressed. So that's an example of an RNA-mediated mechanism of gene regulation. There are other RNA-mediated mechanisms of gene regulation, which is what we're going to look at now. So attenuation is just one. Here is another, riboswitches. Well, let's look at the summary. Before we get there, here. Let's go here. Okay, so other mechanisms, we mentioned attenuation being one, where the leader sequence acts in cis. It's acting in cis. You also have riboswitches, which also act in cis. You have small RNAs or siRNAs, which act in trans. And then you have regulation by antisense RNAs. So we looked at attenuation. Let's look at riboswitches and how they play a role, or how they regulate transcription. Okay. So what is a riboswitch? It says a riboswitch is really a leader sequence with several conformations, similar to what we saw with attenuation. But with the riboswitch, you have two areas of this riboswitch. You have an area, a short sequence, which is called an aptamer, and then you have a second region known as the expression platform. Okay, expression platform. And so the riboswitch is able to regulate both transcription and translation. So let's look at the riboswitch here. So we have the aptamer. Here's our aptamer there of the riboswitch. And then we have our expression platform. Okay. Depending on the conformation of the expression platform, it will determine whether or not transcription will continue. That's transcription of the downstream genes, depending on again, the conformation. Sounding very similar to the leader sequence. Well, it is a leader sequence, but this is a different type of conformation being formed. Okay, so the riboswitch, the conformation that is formed is dependent on what is called an effector. If the effector binds to the aptamer, so the effector binds to the aptamer. When the effector binds to the aptamer, it changes the configuration into one that is a terminator. So this is a terminator configuration, and that allows for transcription to stop. That causes transcription of the downstream structural genes to stop. So the binding of the effector to the aptamer causes the expression platform to form a terminator loop, and that causes the transcription of the downstream structural genes to stop. When the effector is not bound to the aptamer, so here you have a free aptamer, then transcription, the expression platform will form an anti-terminator, and that will allow for transcription of the structural genes to continue. So that is an example of the riboswitch regulating transcription. Okay, riboswitch could also regulate translation. And it does that by making the ribosome binding site available or not. So if the ribosome binding site is available, that's here, see the ribosome binding site is available, then translation will occur. Translation will continue, okay? However, when an effector is present, the effector binding to the aptamer will prevent the ribosome binding site from being available, and if the ribosome binding site is not available, then no translation will occur. Okay, so this is the riboswitch at work. The riboswitch again could regulate both transcription and translation. It regulates transcription by determining whether or not an anti-terminator loop forms or terminator loop forms, and it regulates translation by determining whether or not the ribosome binding site is available or not. Okay, so that's regulation via riboswitch. Okay, let's go back to the list here that shows several forms of regulation of RNA-based mechanisms of regulation. So we looked at attenuation. There's the riboswitch, and then we have small RNAs or sRNAs, and these act in trans. These act in trans. So not only proteins act in trans, RNAs can also act in trans. Okay, so let's look at that. Okay, so these small RNAs, let me make sure I'm on the right side. Yeah, small RNAs, they usually inhibit translation. They inhibit translation by base pairing with the ribosome binding site. Okay, so here we have the ribosome binding site being available to the ribosome, and that would allow for translation to occur. These small RNAs have sequences that are complementary to several messenger RNAs in the cell, not just one messenger RNA, but there are small regions of these small regions of the sRNAs that are complementary to many different types of messenger RNAs. And once these small regions bind to the ribosome binding site, they can bind to the ribosome binding site and prevent translation from occurring. So here we see that in this figure, we have this snRNA, the small RNA, the sRNA, binding to the messenger RNA and blocking the ribosome binding site, and hence it will prevent translation from occurring. So this is an example of negative mechanism of regulation by these small RNAs. These small RNAs can also allow for positive regulation. Positive regulation can occur via these small RNAs. So we can have here translation being enhanced or occurring when these small RNAs, the small RNAs could prevent a configuration that will typically block translation from occurring. So here we have translation being inhibited when the RNA forms a stem loop on itself. You see here is the messenger RNA folding over on itself and preventing translation from occurring. Well, the small RNAs can come in and prevent that stem loop from forming and allowing ribosomes to bind and allowing translation to occur. So these small RNAs can both function. They function both as positive regulators of translation and also negative regulators of translation. Okay, so that's the small RNAs. And finally, another type of RNA regulation is antisense RNA. It's very similar to small RNAs. The difference is that these antisense RNAs are actually made from the opposite DNA strand of the messenger RNA. So we know that DNA is double-stranded, and the DNA in E. coli is also double-stranded. And one strand that actually makes the messenger RNA is called the sense strand. Well, that opposite strand, the opposite strand of the sense strand is called the antisense strand. And that antisense strand, if it's transcribed, could actually block. It could bind to the RNA, it could bind to the messenger RNA, forming this double strand structure, and that could inhibit gene expression. So it's showing you here, the antisense RNA is produced by transcription of the DNA strand opposite of the template strand. So it's called antisense. It inhibits translation by base pairing with the strand. So it could base pair with the sense strand and inhibit translation. It could also lead to degradation of the mRNA, because that base pairing of those two strands could subject that complex, that's the sense and anti-strand complex, could subject it to a special type of RNase, which will degrade that complex. So it can lead to degradation of messenger RNA. And it can also regulate by It says it also interferes with transcription of the sense genes. It's not known how it does it, but it's known that there are times if the anti-sense gene is transcribed, it somehow prevents the sense gene from being transcribed. Okay, so just transcription of the anti-sense gene could block the transcription of the sense gene. So these are that's an antisense RNA is another mechanism of regulation. Okay, so let's see if there are any questions. Okay, so I'm going to say, are there any questions on okay, if not, we'll spend the last 5 minutes looking at using these regulatory mechanisms in the product of genes that are useful, or in studying the regulation of other genes or eukaryotic genes in bacteria. Okay, so let's look at the use of some of these regulatory mechanisms. All right, let's see here. Okay, so the LAGZ gene, we know LAGZ encodes beta-galactosidase, and the LAGZ gene could be used to perform genetic studies of the regulatory region of any gene. So let's say you wanted to know how insulin is regulated, or how the insulin gene is regulated, the gene that produces insulin. Or you wanted to know how the gene for, just think of anything, the gene for growth hormones, how is that regulated? What are the factors that regulate a gene? You could do that by creating what is known as a LAGZ fusion. A LAGZ fusion. You could take the regulatory sequences of the gene that you're interested in and place it upstream of the LAGZ. Because we know what the product of LAGZ is. We know LAGZ produces beta-galactosidase, and we know if beta-galactosidase is produced, we know the outcome of that. Beta-galactosidase breaks down lactose into lactose and galactose, or you could measure the presence of beta-galactosidase by doing the blue-white screening. If you recall, when you did LAGZ produces beta-galactosidase, and in the presence of X-gal, it produces a blue pigment. So you can measure the amount of a LAGZ, or whether or not a LAGZ is turned on or off, by looking at the presence or absence of this blue pigment. Okay, so if you have a gene that you want to study the regulation of, you could take the regulatory region of that gene, so the promoter of that gene, and put it upstream of the LAGZ gene. Okay, so that promoter could be from a eukaryotic cell. It could be from a eukaryotic gene. You could take that promoter, put it upstream of the LAGZ, and then see what factors will turn that promoter on or off. And you could measure that by looking at the blue-white product that is produced when LAGZ, the blue product that is produced when beta-galactosidase is present. So it says conditions that regulate expression of the test regions from the gene X will alter the levels of beta-galactosidase. You see that here. This is gene regulatory region of gene X placed upstream of the LAC-Z, and that could, depending on the conditions present, it could turn the LAC-Z gene on or off. Okay, so that is one use of LAC-Z there. We have 1 more minute. I don't know if we have time. So, well, let's end here, but I think you could go ahead. Well, let's look at another one. I'll skip this one, is easier. Okay, 1 more minute here. We can also use these fusions to overproduce a gene. Okay, you can use a fusion to overproduce a gene. So, for example, we know the lac control regions, which are the promoter site and the operator site. You could take those lac control regions and place it upstream of a gene that you were trying to overproduce. And this is actually done. This mechanism is used to make growth hormones and insulin. You could take the regulatory region of the LAC-Z gene, place it upstream of the insulin gene, or upstream of the growth hormone gene, and we know how to turn on expression of the LAC-Z gene. You do that by, we know how to turn on expression, of the regulatory region of the LAC operon, and you could do that by just introducing lactose into the media. So you introduce lactose into the media, we know lactose acts as an inducer, that will control expression of this gene X, which could be the insulin gene or the growth hormone gene, and that will cause high amounts of that gene to be produced. Okay, I know I rushed through this a bit, so if you have any questions, you could just text me text me or email me, and I'll answer those questions. So that's it for this chapter. It's now 10:21. Okay, so that's it. All right, so that's it. Test on Monday. I'm not sure which TA will be available, but we'll have a TA available for the test on Monday. Okay, have a great weekend. Bye. Have a great weekend.